Hello, I'm Norman Carreck from the UK, and today I'm going to be talking about a European project called INSIGNIA, which is environmental monitoring of pesticide use through honeybees. The INSIGNIA consortium, on whose behalf I'm talking, consists of 16 partner institutions from 12 European countries, and here they all are. And it's a wide range of different skills we have, from modelers, beekeepers, extension specialists, analysts, statisticians, and so on. But before I talk about INSIGNIA, I need to talk about an earlier project that many of us were involved in, which sets the seeds for it. And this project was called CSI Pollen, Citizen Scientists Investigation on Pollen Diversity. And this was done through the auspices of an organisation called Colos, which was originally a very small European project looking at the causes of honeybee colony losses. But since 2008, it's grown to be really the biggest global honeybee research network, with some 1,400 members in 95 countries around the world. And these people are bee scientists, but also extension specialists. And CSI Pollen was set up to explore the possibility that one of the reasons for honeybee colony losses may be that bees were feeding on an inadequate supply of food and a lack of pollen diversity. And whilst an individual scientist can study pollen at one location, the advantage of using beekeepers is you can study many different places simultaneously. So what we did was we got beekeepers to collect pollen on 3 beehives fitted with pollen traps 10 times over the year. Then from those pollen samples, they simply took a 20-gram subsample, enough to fit into the lid of a honey jar. And then they spread them out on a white tray and looked at the number of different pollen colours that were present in the samples. And then they entered the results online using a special link that they were sent. So all the beekeepers were doing was looking at the number of different pollen colours, and we're very aware that the number of pollen colours is not the same as the number of plant species, but nonetheless, if everyone is doing the same thing at the same time, it does provide a good proxy for looking at pollen diversity. And the study took place over 3 years, and in total there were 27 European countries took part, with 586 citizen scientists, of whom 316 were regular enough to use their data satisfactorily. And I was in charge of England and Wales, and I had 71 volunteers. And overall, we collected 17,464 samples. And this shows the advantage of a citizen science project, that there's no way that an individual bee research laboratory would be able to carry out that scale of study. And this is where our beekeepers were. And you can see they were spread throughout Central and Southern Europe, but also up into Scandinavia, Finland, Denmark, and even the Balearic Islands off the in the Atlantic. So it was a very good coverage of beekeepers. And what were the results? There were lots and lots of results, but I'm not going to talk about those in detail. But I just want to draw your attention to these 2 graphs, which are summaries from each of the first 2 years, 2014 and 2015. And these are the number of abundant pollen samples, and this is over the season. And the result was that we found that over all the sites, and in the years, and in places which were very intensive arable and places that were semi-natural vegetation, the bees collected on average 4 different pollen types. Now, they weren't always the same types, but it is noticeable that even in places where there was a huge diversity of different crops available, the bees collected 4 abundant types. And in other places where there was a dearth of pollen and a monoculture of perhaps corn, the bees still manage to collect more than that. And that throws up some interesting points in itself. So at the conclusion to the project, we held a workshop up in Glasgow in Scotland, and we talked through the results. And one of the things we thought about was, well, you know, what should we do with all the pollen samples that we've got beekeepers stored in their freezers and in lab freezers around Europe? Surely we could do something useful with those. And then we thought, well, pesticides are very much the flavour of the month, and this could be a really valuable resource, and we could test these pollen samples for pesticides. And we thought that was a really great idea for about 5 minutes. But then, of course, reality dawned, and we realised that we didn't actually have any control over the way these pollen samples were collected, and there could easily have been contamination from one pollen trap to another, and from one sampling to another. And the major problem being that pollen, being a biological material, decays very rapidly. It's very prone to bacterial and fungal decay. So any pesticide residues in the samples could easily be degraded. So we thought, well, unfortunately, that won't work. And so we gave up the idea. But then we noticed that the European Commission put out a tender opportunity, an advert, for a project they wanted to do called Environmental Monitoring of Pesticide Use Through Honeybees. And they were asking scientists to put together a project to devise a protocol for a citizen science project to get beekeepers to collect pollen for looking at pesticides. And since we had a ready-made protocol for that, we thought that we ought to apply, and we put together a proposal, and were delighted when we were awarded it. And we move on from there. But first of all, I want to think, well, why are honeybees particularly good at monitoring pesticides in the environment? And this is an aerial picture of our laboratory at Sussex University, which is just here in the centre of the map. And here you have the town of Brighton and the English Channel here. And these are the South Downs, which are a range of hills. And we have a little town called Lewes here. And you can see we're surrounded by farmland. And if you put distance rings on there, you can see that there's a range of different places where bees might forage within a reasonable radius of there. And some of my colleagues at Sussex University over the years have done a lot of work decoding waggle dances and then plotting where the bees are foraging at different times of the year. And it really emphasises how far bees can sample the environment. So here, superimposed on a map, is what happens in March, in the spring, and you can see the bees are not really foraging very far away from their hives at all. They're foraging on the campus of the university and a little bit around. But as the season goes on, in April, May, June, July, and August, you can see they're going a lot further. And in August, they are going a lot into the countryside here. There's actually a nature reserve here with very good wildflowers, and also they're going down into the urban area. And you can see in those three areas, there are the possibilities for them to pick up a wide range of different contaminants, from industrial and urban pollution to pesticides and to other things that might be available. In September and October, they forage on ivy and other plants nearby. So INSIGNIA was born, and INSIGNIA stands for Citizen Science Investigation for Pesticides in Apicultural Products. And the aims of the project are to develop a citizen science protocol to use honeybee hive products, principally pollen, to explore pesticide contamination and relate this to the crops on which the bees are foraging. And then we're going to model the risk to bees from different habitats types. So this is just showing the different work packages that are involved in this project. And so we held an inaugural meeting of the collaborators in the Netherlands in November 2018. And we planned that in 2019, which was last year, we would do limited sampling in 4 countries Austria, Denmark, Greece, and the UK, providing a range of climatic conditions, and that at each site we would have, each country would have 4 sites, 2 low risk, that is areas of hopefully semi-natural vegetation, organic farming, nature reserves and so on, and 2 high risk, that is intensive agricultural or industrial areas in each country. And we'd use 3 colonies at each site. And here you can see the locations in those 4 countries. This is Denmark, Austria, Greece, and the UK. And at each of those sites, we asked the beekeepers to collect samples in 4 different ways. So the first one was what we were asked to do, which was the pollen trap, which we had experience of, but bearing in mind that the concerns we'd had earlier about samples decaying or being contaminated still apply, we wanted to back that up with alternatives. And we had 2 alternative passive techniques. We had something called the bee-hold tube, which is a modified entrance that the bees have to crawl through, containing a material which will pick up pesticide residues. We had a novel device called an apistrip, similar in appearance to an apistan strip or a beaverol strip for varroa treatment, but this was coated with a substance that's very good at picking up pesticides, and it just hangs in the hive. And then lastly, we were collecting bee bread samples from the colonies, and this is a novel device that was used to collect the bee bread. And the samples were then split for analysis in Portugal of the botanical origin using a DNA barcoding technique, and then analysis in 2 labs in Spain and Greece working together of contamination by authorized and unauthorized substances. So we were really looking for everything that is an authorized agrochemical or veterinary product, but also things that were known to be used illegally by or unlicensed by beekeepers. So we held a meeting in Spain this year, just before everything came to a standstill, and we looked at the results so far, which I can show you. So these are the apistrips here, and this is just looking at the positive findings in the apistrips in 3 of the countries, and you can see that they picked up a wide range of different compounds, and the interesting thing is that the most commonly found compounds, thymol, coumaphos, fluvalinate, are compounds that beekeepers have put in the hives themselves to control varroa. And there are agrochemicals of various kinds, but they are at much lower levels. And then comparing the different media that we'd used, we find that the apistrip was the best at picking up pesticides, and the pollen samples themselves were actually the least effective and with the most noise in the data. Then looking at the number of pesticides, this is from the 4 sites in Denmark. This was an organic site. This was thought to be fairly intensive agriculture. This is just the first 5 samplings, and we did indeed find that there were more pesticides in the high exposure site and far fewer in the organic area, although there weren't no residues in the organic area, interestingly. And then looking at the DNA concentration picked up in the pollen for the DNA barcoding, we found that the pollen samples themselves were better than bee bread and a lot better than the bee-hold tubes. And this is just looking at the different plant families we were able to pick up in the pollen samples. So some immediate conclusions from the first year were that the apistrips are by far the best medium for detecting pesticides, and that pollen is the best medium for the DNA barcoding studies, and that land use does indeed influence the incidence and quantity of the pesticides that we received. But interpretation of the results is going to be very difficult, and we need to be very cautious about sending back results to beekeepers saying we found X and Y pesticides in your samples, because until the end of the project we won't have a feel for how the findings of individual compounds in the samples actually relates to risk to the bees. So year 2, which is this year, we are sampling in 9 countries: Austria, Belgium, Denmark, France, Greece, Ireland, Italy, Latvia, and the UK. And at each country, we've got 9 sites, also high and low risk, and 2 colonies at each site. And here they all are. So these are being sampled at the moment, every 2 weeks. And a few weeks ago, the first batch of samples were all processed and sent off to the labs in Portugal, Greece, and Spain. And I just want to say, finally, the paper describing the apistrips came out in Science of the Total Environment a few weeks ago, and you can look at that and find out the technique. So this is an ongoing project, so it's very much a case of watch this space. And here is our website that has a blog and many other features. And I just want to thank the European Commission for funding this project. And here are some useful websites. So I will stop there and take any questions you may have.